Welcome back to Offworld, the show where we talk about all things pop culture and space exploration. Today we are talking about Alien Covenant. The whole Alien series always talks about biology and combining different species together, and Alien Covenant is no different. It's a colony ship that's going to a habitable planet, and weird stuff, of course, happens along the way. Today we have two awesome biologists that I'm talking to, so let's join the conversation. Today, I'm with Dr. Jun Axup, a chemical biologist and scientific director and partner at IndieBio, a life science accelerator, and Dr. Lynn Rothschild, an astrobiologist and synthetic biologist at NASA Ames Research Center in Silicon Valley. Thank you both so much for joining me.
Thank you for having us.
Thanks for having us.
So, Alien Covenant, oh my God, totally gory movie. A lot of fun. One of the things that really struck me about Alien Covenant in particular was that in the movie, you have this scene where David is talking about splicing up all of these genes and combining species, or trying to, but it's this very like zoologist scene, and it harkens back to like Darwin, and it's sort of the 1800s, and has like kind of like this old version of like what it means to be like into species and combining species. If they were to be updating his lab to actually reflect where synthetic biology and biotech is today, what do you think that might look like? June?
Yeah. Well, I think laboratories look very similar these days, and molecular biology tools are all very much available and very, very similar. We've been doing this for many decades now, actually, I guess from the beginning of man, of domesticating animals, right? And a prime example would be dogs. They are a single species, but then have been bred over time to reflect all different kinds of different traits that we want. And so now with the new tools like CRISPR and all the other kind of molecular biology tools, such as DNA sequencing, we've really been able to target and edit things. One of my favorite kind of recent speciation kind of projects right now is called, there's a company called eGenesis that is CRISPRing out viruses in pigs to prepare their organs for transplant for humans, because obviously there's a organ transplant shortage, and they're also working on trying to make sure that there's no rejection issues. So things are definitely getting more advanced, and we're just starting to scratch the possibilities there.
Yeah, what do you think? Are there a lot of like interesting tools or things that you would normally see today if someone was sort of like a mad scientist? Would their entire lab be all white and perfectly clean, or like is there just stuff
Labs
everywhere?
are never perfectly clean. I'm sort of on a run with my students to clean every Friday right now, but it is a totally unrealistic situation. Following what June said, we've been actually combining species for domestication for centuries, literally millennia. And the obvious example is the mule. It's a wonderful combination of a donkey and a horse. And so we've done that on the macro scale, even with mammals, for quite a while. Nature's been doing that for quite a while. In fact, there's an alga, friends of mine, these little unicellular photosynthetic organisms, the ancestors of all the plants, that we know about that lives in a hot spring. And the way it's able to do that is it's picked up genes from all sorts of other organisms that have adapted to live there. So its genome is a chimera. It didn't do it because someone told it to or engineered it in the lab. It naturally has that capability. A lot of microbes naturally have the capability to pick up DNA. We actually and other big people organisms pick up DNA from nature using viral hosts and so on. The ability to do this in a surgical way is what is so different today. And I mean surgical, not literally surgery the way you saw in Covenant, but using a very specific methods such as CRISPR and so on. So in answer to your question, the lab, it's a TV, it's a Hollywood lab. You certainly would want to have things like laminar flow hoods. You wouldn't want to have contamination of the genetic material elsewhere and so on. You'd be using very tiny amounts and microfuge tubes and so on. Now, I suppose if you're a robot doing the whole thing, like David or Walter, maybe you're not worrying about contaminating yourself, so maybe some of these precautions that we would use in the lab are irrelevant. But otherwise,
Yeah, exactly. Well, so getting a bit into combining of species. So, in Alien Covenant, he's trying to combine xenomorphs with humans and creating neomorphs, and he's trying a bunch of different experiments with aliens and humans and combining them together. And I know in like biotech, taking some stuff from jellyfish and getting E. coli to glow is sort of like the hello world of biotech. Are we seeing interesting things where people are trying to combine species that I guess go outside of just, interbreeding, that actually go into splicing things together? Is there any examples like that?
Well, if you're trying to get that kind of morphological combination, again, the closest I can think of that's so accessible is the horse and the donkey hybrid to get a mule. It's not actually that easy, and particularly when you're dealing with a multicellular organism. To get there, you have all sorts of pathways, from the three of us going from a fertilized egg to when we were born took a gazillion steps, any one of which, if it went wrong, we wouldn't have been here today. And so when you start to combine organisms that are too far apart, you start to mess up that developmental pathway. So you may be able to get a gene to turn on, but the ramifications may be such that you never make it to a living organism. My other sort of objection, I think, to how this was portrayed in Covenant, and Prometheus too, before Covenant, the prequel, is that everything happens so fast. And I meant to do the calculations before I got here, but this poor woman becomes pregnant, and it's like, this thing materializes in a matter of minutes. And the cell division rate for something like that to happen would be phenomenal, unless you're just talking about very shrunken cells that are just filling up with water. So, there are a lot of things that clearly were done because it was dramatic, but it would be totally unrealistic in nature.
Yeah. And we do all kinds of splicing on the molecular level now, and recombining different genes from different organisms. There's a lot of in kind of like crops trying to make resistance to pesticides and stuff. And yeah, like the GFP is a very great tool, green fluorescent protein. They make like glowing fish now that you can buy. So, it's a big research tool used as well. But at the same time, understanding where you're putting that DNA, how you're getting it in there, making sure all the regulations correct, making sure that protein doesn't interact with other proteins in the organism. That all is very complicated, and a lot of it right now is trial and error, and that's why PhD students spend 5-6 years working on these projects, because there is a lot of kind of brute forcing and kind of seeing what comes out the other side.
Yeah, I mean, so something that has come up in conversation recently has been about, if CRISPR or other techniques can actually be used on humans, and some people have, debated if it won't work on humans. Are humans inherently harder to be making modifications to than, tiny little microbes? Is there something unique about humans, or is it kind of the same process?
I don't think the problem is that it can't be used on humans. The problem is that you can get off-target problems. So for people who aren't aware, CRISPR is like a really incredibly good molecular scissors. So if you had a book and you told someone to take a pair of scissors and just cut it randomly, you would get a mess. But if you said you need this string of 26 letters and you can only cut there, you would have very specific places. And that's the beauty of CRISPR. It's like that, taking 26 to 30 letters and only cutting at those places. And so that's great for the vast majority of things. The problem is it's not 100% perfect. So sometimes you get an off-target mutation. And if you're working on a bacterium to try to make it glow or make more some kind of chemical or more resistant to radiation or whatever, so you get a little off target. Maybe you say, okay, I'm going to throw away that test tube or autoclave it, and that didn't work, or we can tolerate that. But it's very different for a human. So it turns out you have an off-target mutation and you've now caused some huge genetic defect, something like that, or this poor kid's born without a liver, or who knows what. And that, I think, is part of the whole risk of using on humans, is unintended consequences, just because CRISPR is an unbelievably good pair of scissors, but no pair of scissors is perfect.
Yeah. I would also add that the off-target is definitely the major issue that people are worried about. But CRISPR at the moment is actually not very good for larger species and larger genomes. Like, so we have 3 billion genomes, or 3 billion base pairs in our genome, and so the likelihood of having multiple sites that it can cut is part of what Lynn was talking about. Furthermore, it's easy to cut something out. Well, relatively easier to cut something out of the genome, but it's really hard to put something in. And then that efficiency right now can even be like 10% or less, and people are working on tools to get it more robust, more targeted. Delivery is also an issue. So even if you have the CRISPR scissors, how do you bring it to the right cells at the right time? So those are all very open questions. But I believe we will be able to get those tools.
Right. I think what we're both saying is there's nothing inherent about humans that is going to preclude using CRISPR at some point. It's just I don't think we're ready to yet because of the unintended consequences.
Got it. Yeah, so another thing we see in the movie was at some point the crew is walking around this new eerie planet, and they step on some, things on the ground, and they produce airborne spores, and these spores like go into the guy's ear and implant in the ear, and then before you know it, he's getting infected with this alien, inter-alien species thing. Are airborne spores like a good method for microbes to find, incubators? Is being airborne as a microbe like a benefit, or is it a hindrance?
Oh, it's brilliant. I mean, think of all the diseases like the common cold and chickenpox and so on. Those are all carried by airborne spores. They've been very, very successful doing that. Plus, there are a lot of microbes that are able to transmit actually worldwide. There was a wonderful study a couple of decades ago in a pond in England, and they basically were looking using different culture media for almost anything, and they were able to find much of the world's population of microbes there because they're all over. There's almost no such thing as an endemic microbe. But it really is better than that because some spores are unbelievably resistant. So I've been pushing my lab at NASA to use Bacillus subtilis a lot. It's a common soil microbe, and therefore it's designed to have a really resistant spore. And when I say really resistant, Bacillus subtilis spores were actually flown on the long duration exposure facility, LDEF, a couple of decades ago for nearly 6 years. And this exposure facility allowed them to be exposed to space vacuum and radiation and so on, and yet there were Bacillus subtilis spores that survived almost 6 years just out in space.
Wow.
So that is absolutely the record, and so I figure it's good enough for them, it's good enough for us. We have a satellite mission that we're preparing for. We've had the samples actually in the flight hardware now for over 2 years, which would normally be a problem, but we've been checking periodically on ground controls, and darn it, they're not at about 99% survival. Bacillus subtilis spores are really tough. So this whole idea of using a spore or things like cysts, which are very similar, or even a seed, which most people are familiar with, some of these have been, shown to be able to survive literally millions of years. So it's unbelievable. How do we know? They've been preserved in amber, or people have taken spores and seeds off herbarium samples, which are very, very carefully marked with whoever collected it that day, and so on. So it's an incredible way for organisms to persist and to propagate and to distribute.
Yeah.
Think of pollen.
Yeah, exactly. And that's why in the laboratory we have to autoclave everything. So that's 120 Celsius and 100 kilopascals in order to try to kill every single spore. Because bacteria, once they get into a condition that's not ideal, they will germinate and become a spore to try to survive. And so it's a very robust way to increase to keep their species alive.
Yeah.
Cool little fun fact actually I learned this morning was when you breathe in spores, there's actually a triage as you go through your lungs. So the bigger ones are going to hit with a certain velocity, the sort of things we're used to at NASA, worrying about particles hitting impactors in space. It's exactly the same thing when you breathe in. So the larger spores are going to hit your nasal cavities and areas of your mouth and so on, and the little smaller ones will get further down, and the little smaller ones don't impact, but they can get further down. And so even as you take these spores in, whether it's through your mouth or your nose, usually not your ears, unless it's in the in the water, you're actually doing a separation just because of impacts. Again, it just really rang with me because it's exactly the same as worrying about impacts from micrometeorites and so on.
Yeah, so I mean, so these are some like bad examples of microbes that future humans could encounter on alien planets in sci-fi. What are some examples of like synthetic biology that might be really useful for us to take into space with us, that either for terraforming or for just human longevity or anything?
Yeah, I mean, you mentioned bad microbes, but we also have a lot of good microbes, right? All in our guts and on our skin, protecting us. They are part of our immunity. They help us break food down. So yeah, there's definitely a lot of good things there too. And things that we can take to space, exactly. The terraforming, we are realizing more and more that so much of our soil is covered in all these spores and fungi and all these different organisms. People are already starting to do engineering on these microbes to have them increase nitrogen fixation to deliver more nutrients to our crops, or seed coating, putting microbes on seeds to help them germinate. Eventually microbes can help with air, water, and the soil. And those are kind of fundamental for terraforming.
Useful for space, yes.
Well, actually, NASA realizes it's so important that about 10 years ago I was asked to start a program looking into that very question. It's using synthetic biology to enable space exploration. And I think one of the biggest deals is that once you start looking at biology as a technology, it can do all sorts of things that normal technology with an on-off switch can't do. So, for example, biology is self-replicating. I mean, you don't have self-replicating computers or robots or anything like that right now. Life is self-repairing. Life can go dormant in spores, like we were talking about a moment ago. Life can use sunlight as energy during photosynthesis, for example, to make more of itself. So if you start to, and not to mention chemical production and assembly, life is the best nanotechnologist that you're ever going to find. You know, every cell in your body is a better nanotechnologist than anyone with all sorts of fancy degrees and awards, because you have to have atom after atom precisely laid down, or your cells can't function. So if you take that power that life has and use that off planet, you're able to solve a couple of problems. One is the upmass problem. So you engineer organisms so that they're happier living on Mars. Maybe they're more radiation resistant. Maybe they're better at growing at a little bit lower gravity. Maybe they do all these things, increase nitrogen fixation, use different parts of the solar spectrum or whatever. And then you get them to live off the land. So you get organisms, for example, that can use the sunlight and the CO2 in the atmosphere, just like on the Earth we have cyanobacteria, we have algae, and now we have plants that are that interface between the raw materials on planet Earth and the rest of life on Earth. And so it's just sort of adapting what's happened on the Earth, make it more efficient for the environment off planet and for our needs, and that is really going to be the solution to long-term settlement anywhere.
Yeah, well, one of the things that I like that I know you've worked on that is kind of outside of direct life support has been making microbes that could chew up electronics and then produce copper as a result, and so that we don't have to worry so much about bringing a lot of electronics that we can't recycle with us.
Absolutely, and it's more than recycling. Up in space in the hard radiation environments, the life cycle of electronics isn't as good as you would want it to be. And so where are you going to get new ones? I always sort of joke, they don't have overnight delivery with Amazon.Mars yet. Maybe someday they will, but they don't at this point. And so you have to be able to live off the land. You have to be able to bring things that are low up mass, because it's very expensive to launch things into space, and that are flexible and reliable. And so the project that we've been working on is actually taking organisms to grind up and digest, well, not really digest, but separate spent electronics. And so the idea is that many of the proteins in your body actually bind metals, over 50% of them. And so if you take these parts of the proteins that specifically bind metals, you engineer those into a bacterium, which is what we've done, actually onto the surface, and they're then very specific for pulling out metals. So, for example, you grind up the electronics, you have a bacterium that is specifically designed to bind copper. or zinc or nickel or whatever. You put them in this mix, and the idea is that they then have these atoms of zinc or copper or nickel or whatever attaching to their outsides. You then pull the microbes off, and you can then take the metals off, or in our case, what we did was we used plasma printing, which actually vaporized the organisms and left with the metals. So it was an end-to-end biological recycling system, which, by the way, then has huge spin-offs for the Earth.
Yeah, totally awesome.
And in addition to microbes, there's going to be so many other problems, right? There's the planetary health, but then the human health side as well. There's going to be food scarcity. How are we going to be able to grow all the food that we currently have, or do we have to go for other things like lab-grown meats, which are companies that IndieBio is sponsoring, and kind of more synthetic alternatives. There's 3D printing organs and tissues, regeneration, obviously lots of therapeutics, and we don't know, our bodies will be completely different in space. The stresses, the changes in microgravity, we're not sure how that's going to happen. So we need to develop more tools and do more studies on Earth now to figure out those potentials.
Yeah, absolutely.
And I think you touched on something that we can do the studies on the Earth, but that ultimately will not give us the answer. And that's been the beauty of having ISS up there, and Mir and the other space stations, and the, thank you for the astronauts and the cosmonauts and so on who have spent long periods of time over years on these orbiting labs, because then you get some idea how a few individuals have reacted. Now, you need a lot more. But the fact is that you can prepare people for space, till you're blue in the face, till the cows come home, whatever metaphor you want to use. But the fact is that everyone's going to respond a little differently. And so you want to be able to deal with that on site. You can't do everything on the Earth. And so whether it's a microbe that you want to send to sort of a microgravity finishing school once you're there, or astronauts, once they're there, you may need to check their gut microbiome or their surface one and say, you know what, we need to tune this up a little bit. Right here and now. And so that's, again, the beauty of using something like synthetic biology and biologically enabled technologies, because you ultimately should be able to do these things on site.
Yeah, absolutely. Yeah, I totally love the interplay between Earth and space, and how space can help Earth and Earth can help space, and I just love it. So in space, we can't really bring a ton of stuff with us, or it's very expensive to bring a lot of stuff with us. How do we get around that, or how are we tackling that?
So let me give you part of the dream of the future. And that is that you have a DNA production facility on Mars or the Moon or ISS or whatever. And so you're actually able to make genes and bits of life forms off planet. Now, if that sounds crazy and futuristic, you have to stand back and understand that all of us and our students every day are in their offices coming up with DNA sequences on their computer. They're hitting send. The information then goes to a gene synthesis company, and they then send back to the physical DNA. So it really doesn't matter once you've made that break between the information and the actual substance of life to have the information sent off planet. I mean, it's no different than having it sent down the street to your local company. And so we now have that capability to come up with that information on the Earth or ISS or whatever, and send that information to Mars or Moon or maybe back to Earth to then actually recreate these genes and put them in another organism. And so that's an incredible advance, an incredibly important point.
Yeah, Jun, with IndieBio, I know that you're taking in a ton of startups who are doing a bunch of different things, and a lot of life support systems might actually become lighter because they're using microbes. Is that true that like a lot of microbes are actually replacing machines that maybe are big and bulky, and instead we've just got little bacteria that are just doing a lot of the work of like heavy things?
So yeah, microbes are great at producing, like Lynn mentioned earlier, you can program them and essentially to make chemicals. And so you can make all these chemicals at mass so that you don't have to bring all those materials. So yeah, with like 3D printing organs and growing up cells and everything, it's going to be much easier to create in space than to have to bring all those things.
Yeah, absolutely. So my final question that's really, I guess, meant to be more of a fun question is, in a futuristic timeline where we can combine humans with another species that could help prepare us for some aspect of space travel, which species would you like to combine with humans to get some of their superpowers? Jun?
Yeah. My favorite species is axolotl. So it is this Mexican salamander. And from what I understand, it's actually kind of an intermediate, kind of a like a teenage salamander that kind of got stuck in that teenage age.
Okay.
And so it regenerates its limbs if you cut them off. It doesn't have cancer. So it actually, this DNA was sequenced recently, 30 billion base pairs, so 10 times more than ours. And people are starting to dive into that to understand what makes it tick. And so maybe we can fuse a little bit of those genes into ours and hopefully regenerate our limbs in space.
That'd be very cool. Lynn, do you have any?
Well, I
favorites?
don't have a clean answer for you because my background's in evolutionary biology, and so I look at the world and say, this is a fabulous diversity. How did we get to this point, and so on, and isn't this marvelous? But as a synthetic biologist, I look out there at this diversity, and it's like this giant genetic hardware store. And so I can think of genes from one organism I'd like to add so that maybe we've got more resistance to radiation, and something else so we have more resistance to oxidative damage, and maybe something else so our guts digest better, or whatever. And so it's this mix and match that I think has huge, huge potential.
Absolutely. Well, thank you both so much for joining me today.
Thank you.
Yeah, thank you.
One of my last questions was about what we would combine with humans to explore space better. Animals, plants, bacteria. What sorts of weird things would you combine with humans so that we could be better space explorers? Thanks so much to my two awesome guests. That was a great conversation, and I want to hear from you in the comments. And remember to subscribe and like on YouTube, and I'll see you next time.